Hi, my name is Kate Clark, and I'm the Technical Director at Microbial Insights. We appreciate you taking time to join us today. Microbial Insights is delighted to have speakers today from a great company that does a lot of work to help mitigate corrosion, DNVGL. Joining us today are some amazing scientists, Rick Eckert, Susmitha Kotu, and Christopher Kagarise, who will be discussing laboratory testing of biocides simulating field conditions. Rick Eckert is the Senior Principal Engineer in the Corrosion Management Group with DNVGL and has been involved with pipeline corrosion and failure investigations and forensic corrosion engineering for over 35 years. His recent focus has been on internal corrosion management and optimization of mitigation and monitoring programs. He's a recognized expert in microbiologically influenced corrosion, and he's certified as a NACE internal corrosion specialist and currently serves on the NACE Board of Directors as Publications Chair. He received the NACE Presidential Achievement Award in 2004 and the Distinguished Service Award in 2014. Dr. Susmitha Kotu is an engineer in the Corrosion Management Group, and her expertise is on understanding microbiologically influenced corrosion. Susmitha received her PhD in chemical engineering from Texas A&M and worked on projects with ExxonMobil and Baker Hughes before joining DNVGL. Dr. Christopher Kagarise is a senior engineer in the Corrosion Management Group, and his work has focused on helping oil and gas operators to improve the internal corrosion in their pipelines and develop corrosion management plans. Please note that this webinar is being recorded and will be available in the resource section of Microbial Insights webinar within the next week. A link will also be emailed to everyone who registered for the webinar, so there's no need to contact for that. After the presentation, we'll have a short question and answer session. Please feel free to type your questions throughout the presentation, and our speakers will answer as many as possible at the end. Without further ado, I'll turn it over to our speakers.
Thank you, Kate. Thanks for the opportunity to be here with you all today. I hope if you're in the U.S. somewhere that you are staying warm and safe in the midst of the conditions that we're facing. Today we're going to be talking about laboratory testing of biocides, simulating field conditions, to talk about some of the work that we're doing in our Columbus, Ohio laboratory. Chris, let's go to the next slide, please. So we want to talk a little bit today in this slide presentation about MIC in the oil and gas industry, just some of our general experience in that area. We want to focus on the application of advanced molecular microbiological methods. How are we using these new methods in terms of biocide selection, failure analysis, and other problem solving that has to do with microbiology in the oil and gas industry. And we're going to focus today on some of the work we've been doing in testing of biocides in the laboratory under simulated conditions using biofilms that are grown from actual field samples. Okay. So DNVGL, overall, we're consultants to the oil and gas industry, and the team you're speaking with today, we are, Chris, Smith, and myself, we're involved in internal corrosion and MIC very often, and in areas such as bioremediation as well. We work with a global cast of colleagues in UK and Denmark and Norway and Singapore, South America, that reach out to operators in these other areas. So our work kind of takes us around the globe in terms of MIC. So that really we get into some interesting challenges that are faced by different operators. We're not just in the oil and gas, so DNVGL is also into energy systems now. They have been for quite some time, and we also have had a very large presence in marine certification and verification services. So been around a very long time. I guess our just some context for today is that we want to talk about how basically a practical approach for laboratory testing of biocides, using better screening technologies than what are currently available in some of the NACE standards. And this is an area that is certainly growing, the use of biofilms for biocide testing. Susmitha is actually chairing a NACE/AMP committee that's working on a standard around that area. So you have the right people. Chris? So Microbial Insights, DNV has been working with our friends at Microbial Insights for well over a decade now. We value their high reliability and technical integrity and molecular methods. They've supported us on many MIC forensic investigations, our biocide testing experiments, and many of our laboratory mix studies. And we've also teamed up together on various projects too. We found that they have expertise in many areas besides molecular methods, and they've been an enormous help in logistics and transport and getting samples from all over the world back to the US to where we can do our work with them. So we very much appreciate them, and we, again, thanks to Dora and Kate and their team for inviting us to present this today. And now I'm going to turn it over to Chris.
All right, thank you, Rick. Thanks everyone for joining today. So just to give a little background then on our further background on our talk, I'm sure if you've joined us today, you're familiar with MIC in general, but if you're not, this is just a brief overview of kind of what we see in the oil and gas industry, kind of where we're coming from with our testing and what we're going to talk about today. So, you know, we're focused on microbiologically influenced corrosion, and the issue is that microorganisms are everywhere. So if you look inside of a pipeline, outside of a pipeline, anywhere on Earth, there's a good chance you're going to find microorganisms. And so that becomes a problem for us when we try and figure out if corrosion is due to microorganisms, because you're probably going to find them. The tricky thing though is not whether you find them, it's what are they actually doing something to cause corrosion? So that's one of the places where this the techniques we're going to share today come into play, and some of the evidence we gather to try and support whether microorganisms are doing something to influence corrosion on the pipeline or not. We have these microbiological methods that help us determine what's present, if they're active, different types of species that are present. We have more traditional methods like culture testing that can tell you how many of a certain type of microorganism are present. But the thing that those tests aren't able to do is there's no clear correlation between numbers or types of microorganisms and whether you have corrosion and what the severity might be. So say you find 10 to the 6 bacteria present on your pipeline, that doesn't readily correspond to a corrosion rate. So that's one of the more difficult challenges with MIC, is that it requires a lot of evidence and testing to determine if your environment, your microorganism population is contributing to corrosion. So we'll talk about that a little bit here in the next few slides. Then one of the key things to keep in mind too is that when it comes to MIC, what's most important is the biofilm that grows on the surface of a pipeline. So we're interested mainly in pipelines in our work, but this is true of any sort of structure or item that's suffering from corrosion. But we want to know what's present in biofilms. So you can do tests on, say you have a fluid like oil or water that's being transported through your pipeline. You can see what's floating around in your pipeline. So what microorganisms are floating around, those are your planktonic bacteria. Those are important, but what's really important is what's happening on the surface. So that's where a lot of our effort is focused, is on looking at what's growing on surfaces and how biocides or other mitigation efforts influence those biofilms. So talked about this briefly. Where is MIC found? Short answer is basically everywhere in the oil and gas industry you can find it. So if there's water present, and microorganisms present, which we've already said they're basically going to be able to find them anywhere, there's a potential for MIC to occur. So there are a number of different factors that can contribute to MIC. If you have low flow or stagnant flow, that can contribute to biofilm formation. Certain operating conditions, pressure and temperature ranges, pH ranges, microorganisms can thrive in a wide variety of environments. So it's hard to have an environment where MIC would be excluded. But in general, most operating conditions you're going to run into in the oil and gas industry enable or would allow for MIC to occur. So when we think about MIC and how we manage it, we think about it in kind of this three-step process. So the first step is we want to assess the threat. So what we want to do is determine is MIC a legitimate corrosion mechanism? So there, you can have electrochemical corrosion where there's things like acid gases, CO2 and H2S present, or you can have oxygen present. Those conditions can contribute to corrosion, and those are independent of MIC. So if you want to know if you have a corrosion issue in your pipeline, you want to know what to treat, you need to first determine what's causing the corrosion that you're seeing. And this can happen, it can change over time, conditions can change, so rarely do you run into a steady state process in pipelines. So things are always in flux, always changing. So it's important to think about what conditions you have and what corrosion mechanisms may be active. Once you've identified what your corrosion threat is, you want to determine what barriers or mitigation can you apply to minimize that corrosion. So one example is you can use chemical treatments. Say you have MIC as a problem and there's microorganisms, you can use biocides, or you can use pigging to mechanically clean your pipeline. So there's a number of different mitigation options that you can tailor towards your specific corrosion mechanism. Then once you've applied your mitigation, you want to determine what effect it's having. So that's measuring something in the field and monitoring the process to make sure that conditions haven't changed or the mitigation you're applying is actually having a positive effect on the corrosion rate. So if you're going to pig continuously, you want to make sure that it's doing something to influence the corrosion rate, so you can see that benefit for the action you're taking. So I kind of mentioned this earlier, but when we think about MIC and whether it's occurring or not, we don't rely on any single piece of information because there is no single piece of information that can tell you if MIC is or is not occurring. So we look in 4 general categories of evidence. There's the microbiological environment, so that's things like the pH of the system, temperature, what microorganisms are present, how many are present, what's their activity. We look at the chemical environment, so what anions, cations are present. Is there something else that can be contributing to an electrochemical mechanism? We think about the materials and corrosion products that are present. If you see iron carbonate present, that can tell you that you might have a CO2 corrosion mechanism that's occurring. So we want to think about those clues that we have. And then we also want to look at design, operation, inspection of this system. So, where is corrosion occurring? Is it happening in certain segments that operate under certain conditions? Is it happening just on the bottom of a pipeline, the top of the pipeline? Does the pipeline flow continuously or is it static? So those are the sorts of questions that we consider and try and piece all of these things together to determine whether MIC is or is not an active corrosion threat. There's a lot of ground we can cover here with this, so that's just kind of a brief introduction. I know we've given seminars and webinars just on this topic alone. But hopefully that gives you some idea of the different things that should be considered when thinking about MIC and whether it is or is not a relevant threat. Just to briefly mention, we said, there is no easy one test that you can perform to tell if MIC is or is not a threat. We're going to talk about this a little bit later at the end of the presentation, but there is a JIP that's being planned to identify biomarkers that may be able to help be a simple test that can help identify if MIC is or is not occurring. So that would be a pretty big change to the current state of technology and enabling people to better determine if MIC is or is not concerned. But again, we'll touch on that later in the presentation. So now I will turn it over to Susmitha.
Thanks, Chris. So in the next few slides, I will cover different microbiological methods that can be used to understand whether MIC is an active threat in the system, compare which biocide is more effective in mitigating MIC, and whether MIC is the mechanism that caused a failure or a leak. So some important questions that you should be asking when investigating microbial communities and whether they're causing MIC is how many microorganisms are present, who is present, are they known to contribute to MIC, and whether these microorganisms are active. There are various methods that are used to determine these microbiological characteristics, and these methods have evolved from relatively lower priced culture-based methods, which is mainly serial deletion tests or bug bottle tests, to more expensive DNA-based methods like qPCR, quantitative polymerase chain reaction test, or NGS, next generation sequencing, and more quicker enzyme-based tests like ATP test, which stands for adenosine triphosphate assay. Next slide, please. So we'll first start with culture-based methods. Serial dilution tests are the most commonly employed culture-based methods, and they're primarily used to identify whether sulfate-reducing bacteria or acid-producing bacteria are present, and if they are present, how many cells of these bacteria are present. And these tests help answer the questions on whether these groups of microorganisms are present and how many cells are present. And because microorganisms in the samples that you're collecting are grown in nutrient media, which is usually put in these bottles that I have here on the slide, because microorganisms are grown in this nutrient media and they're incubated for a certain period of time, it's very common to see false positives and false negatives with this method, and it's not very reliable. Next slide, please. So the next method I'll cover is a DNA-based method, quantitative polymerase chain reaction, qPCR. So qPCR is used to measure the abundance of bacteria, archaea, and different metabolic groups of microorganisms, and that is not restricting just to sulfate reducers and acid producers, but also other groups like methanogens, iron-reducing bacteria, metal oxidizing, and sulfur oxidizing bacteria. And this method identifies and quantifies microorganisms that cannot be cultured in liquid media. And when you use samples for qPCR tests, you can use both planktonic and biofilm samples. And this method tells us how many total bacteria and archaea are present and whether these important metabolic groups of microorganisms that have an association with MIC are present. And based on the metabolic groups that are present, we can infer whether there's a possibility of MIC occurring in the system by looking at what is present and the numbers of the different metabolic groups. Next slide, please. NGS, or next generation sequencing, is another DNA-based method that is used to assess the diversity of the microbial community, but you do not need to have any prior information of which microorganisms might be present. This method identifies microorganisms but does not quantify it. It only gives you like relative abundance, but not like absolute numbers of what might be present. And again, this method can be used both with planktonic and biofilm samples after DNA has been extracted. and a universal gene known as 16S rRNA gene is sequenced and compared to a database to identify the microorganism to the genus level. This is useful in interpreting microbiological information on what is missing from culture and qPCR data. And both the DNA methods that we discussed here, qPCR and NGS, give most reliable information on who is present, as in which microorganisms are present. The other important factor to consider while testing for microbiological environment is the activity of microorganisms. Microorganisms might be abundantly present, but if they're not active and are in a dormant state, they will not be involved in corrosion processes. Hence, it is important to check if microorganisms in a sample are active, as only if they're active, they can be contributing to corrosion. So to test for microbial activity, we use ATP assay. ATP is an energy storage molecule present in all living cells, and only microorganisms in an active state produce ATP, and hence a measurement of ATP is an indirect indicator of microbial or biological activity. Microbiological characteristics vary between planktonic and biofilm samples, and because MIC is primarily mediated through biofilms, microbiological analysis from swabs or biofilms gives the most information than analyzing liquid samples or planktonic samples. So the type of sample used for testing determines the confidence in the data when we are trying to piece together information on whether microorganisms are causing MIC or if a particular biocide is more effective against compared to another one. So to summarize, whenever microbiological testing is being performed, biofilm samples should be used for identifying which microorganisms are present and to answer the questions on which microorganisms are present and are they known to contribute to MIC. The most reliable methods are NGS DNA sequencing and qPCR. And to identify how many are present and to get an abundance of the different groups that are present, qPCR gives the most reliable information. And to see what the activity of microorganisms is and to see if microorganisms are active and whether they could be causing corrosion, ATP assay gives us the most information. And as Chris described earlier, data about chemical environment, material susceptibility, the corrosion products that are forming, the system design and operating parameters should all be used in conjunction with the microbiological data that we collect with some of these methods that I mentioned for MIC investigations to understand whether the environment is capable of supporting microbiological growth. Now, Chris will discuss a mitigation strategy for MIC.
All right, thank you, Susmitha. So this is kind of an outline for our general mitigation strategy that we use when we work with various clients. So the first step you can see here is we want to fully understand the type of threat we're dealing with, whether that's MIC or other types of corrosion mechanisms. You want to understand the severity. So if you don't know how bad the problem was before you started mitigating it, it becomes more difficult to show that the activities you've performed. Susmitha, is my screen showing the right slide? Sorry, I had a glitch.
No, yeah, now you're on the right slide.
Okay, sorry, I'm not sure what happened there. So you want to understand the threat, so whether it's MIC or electrochemical corrosion threats, the severity, so that when you apply your mitigation, that you know you've had an effect on corrosion rates, or number of internal corrosion anomalies that you found, or anything like that. So you want to fully understand that, how bad it was before you apply, so that you know that you've made an effect after you've applied your mitigation. You also want to know where your corrosion problems exist, again, so that once you've started mitigating, that you can focus on those areas and make sure that they're getting better, and also that you're not also creating a problem somewhere else. Sometimes we have seen where you apply a chemical, and that can have unintended consequences. So it's very important to know what's going on both before and after you start your mitigation program. Once you know what problem you're going to address, what your corrosion mechanism is, you can evaluate what different possible mitigation measures you can use. So if you have the potential for MIC, usually that leads you down the road of using biocides or mechanical cleaning. If the corrosion mechanism is some kind of acid gas corrosion like CO2 or H2S corrosion, maybe it involves using a corrosion inhibitor. Sometimes both of those mechanisms can occur at once, and so you may need to use multiple different types of mitigation chemicals or methods, gas removal strategies. So there's a number of different strategies you can use, and it all depends on knowing what problem you have that you need to treat. And so we'll talk a little bit more about how to evaluate biocides here in the next few slides. But then the next step after that is implementing your mitigation, so taking that out to the field and seeing what kind of effect you're having, and then reviewing again once you've applied your mitigation, collecting data, reviewing if it worked, and making changes as needed based on the results that you're seeing. So, when we select the method, I talked about this briefly, you should base it on what you know the applicable threat is. So if you're unsure what your mechanism is, if you think if it's MIC and you're applying a biocide, but the corrosion mechanism is really H2S corrosion, applying biocide may do nothing at all, or it may make your problem worse. So again, knowing what you're dealing with to start with is the key. And as we talked about, the way we try and figure out with our clients, other operators, how what corrosion mechanisms are active or what threat you're dealing with is by collecting multiple lines of evidence. So the operating conditions, using our microbiological methods to look at what populations of microorganisms are present, their activities, the things that Susmitha discussed, and build that story to see what threat we're dealing with. And again, so it can be any number of different factors that contribute to the corrosive conditions that we're dealing with. It's not necessarily just MIC or just electrochemical corrosion, can be a combination of the two. So say in our example, we've determined that MIC is a threat and we want to look at different biocides. So how do you go about selecting the right biocide and making sure that it's going to be effective? So the way we generally recommend going about this is first performing some laboratory testing for effectiveness. So we'll discuss different methods for doing that. Then once we come up with a lab validated method, take that into the field and apply that and look at what happens once it gets into the field. Because you can't simulate everything in the laboratory exactly as you might expect in the field, and conditions change in the field. So you want to make sure that what you've evaluated in the laboratory transfers to the field. And then the final step there is kind of optimizing your treatment method based on the results you're seeing during your normal operations. And so when we design laboratory tests to look at biocides, there's kind of 4 basic categories we think about when we design a test to evaluate a biocide or a chemical. So first, we want to think about selecting a representative system. So for example, if you want to treat maybe hydrotest water during hydrostatic testing, that would be different than if you're treating a pipeline that transports crude oil. So the way that biocide could be applied in those cases would be completely different. The operating conditions are different. So the conditions are different. So first you want to think about, what does your system look like? How does it operate? What's the operating temperatures and pressures, and try and be able to control as many of those variables as possible, and make the system that you design as close as possible to the field as you can. Second, we talk about compatibility testing. So you want to think about if there are other chemicals, treatments that are being used in your system. Say you're currently using a corrosion inhibitor, or scale inhibitor. You would want to consider looking at the use of the biocide in the presence of those other chemicals to see if there's any negative interactions between them. So that's what we're getting at there with compatibility testing. Third, concentration and dose. So with every chemical, there's a wide range of concentrations that you can look at. So a lot of times we look at applying, something in the low, medium, or high category, or a spectrum of different concentrations to see what's kind of the minimum effective dose, or how does corrosion and microbiological behavior change with different doses or concentrations. The other thing you can think about too is whether you're going to apply a chemical on a continuous basis, or if it's going to be a batch basis, or something in between. So how can chemical be applied in the field, in the real world, and can we replicate that in the lab? And then finally, effectiveness testing. We'll talk about this a little bit more, but when we look at biocide effectiveness in the lab, we're looking at both the ability to influence the microbiological population, their activity, what's present, and also corrosion rates. So that's kind of a key thing for us too, because ultimately that's what you're trying to do, is minimize corrosion. So we want to make sure when we're testing things in the lab that we're, in addition to everything else, we're looking at what happens to corrosion on samples of pipeline or steel or whatever we're testing. So that's something important to keep in mind. So kind of touched on some of these, but some more test parameters we can think about is one of the key things here is length of testing. Sometimes, you may be dealing with a system that's going to be exposed for a short period of time. So say you're doing a hydrostatic test. Water could be in your pipeline for a week, and you want to see, is there a chemical that's effective at minimizing the potential for MIC or corrosion for a period of 1 week. That would be different than, say, the crude oil pipeline that's in continuous operation and is going to be operating for years on end. We've seen lab that microbiological populations change over time, the way biofilms form or influenced by chemicals vary over time. So you want to think about that aspect as you design your test, whether you can evaluate for a couple of weeks, or if you need to move to a longer term test. We talked a little bit earlier about field conditions. So again, we want to try and create a test where we're operating under similar temperatures and pressures, because that can influence what microorganisms are growing or forming biofilms. Thinking about flow rates and shear stresses at surfaces, because that can also influence biofilm growth and density. We want to think about dissolved gases that are present, if there's CO2 or H2S or oxygen present, because those can influence both corrosion rates in general, but also the way microorganisms behave. Thinking about different hydrocarbons that might be present, if there's crude oil present or natural gas, whether we have aerobic or anaerobic conditions. So all of these things we want to think about and try and simulate as closely as possible in the field, so that we get the most accurate representation of our system while we're doing testing in the lab. And again, biocide application, typically you see either a batch or continuous application. So think about how that could be applied in the field and whether it makes sense to do one or the other and simulate that in the lab. So I'll turn it back over to Susmitha to discuss some more testing and how we use some various results.
Thanks, Chris. So in this slide, I'll be covering sample selection and characterization. Sample selection is an important aspect when performing lab testing. Use of process fluids from the field is preferred when possible compared to using like stock cultures. Stock cultures are unlikely to contain like the same distribution of microorganisms as process fluids, and microorganisms tend to behave differently when different members of a population are added or missing. Therefore, true effectiveness of a biocide is best evaluated on a sample by using process fluids, which represents the true conditions of the field. Also, it is very difficult to replicate fluid chemistry when performing these tests in the lab. Process fluids contain chemicals that are used for treatment like corrosion or scale inhibitors, drag-reducing agents, oxygen scavengers, and all of these also influence the effectiveness of biocides. And hence, using process fluids can help simulate the chemical and microbiological environment as closely as possible. So once a sample is selected, the chemical and microbiological characteristics of the sample should be characterized. Chemical composition of the process fluids can give us important information on chemical indicators that could have either been formed as a result of microbiological activity, or these chemical indicators would be providing nutrient sources for microbial growth, or there could be abiotic conditions that can maybe explain the corrosion mechanism that's happening. Chemical composition helps characterize pH, alkalinity, if there's any dissolved solids present, dissolved gases like H2S, carbon dioxide, the salinity, and if there are any carbon sources, any organic acids that I mentioned that could be nutrient sources or maybe byproducts of microbial metabolism. There could be electron donors like acetate, lactate, hydrogen, sulfur, and iron, or electron acceptors such as nitrate and sulfate. So all of this is important, as I mentioned, to help understand if there are any indicators of microbiological activity or if there are indicators of abiotic corrosion mechanisms. Microbiological characterization should also be done before the start of any test because that gives us a baseline of looking at what microorganisms are present, how many are present, and what the activity of these microorganisms is at the start of the test, and the similar characterization can also be performed towards the end, and that can be compared to see how effective the biocide was in not just changing the overall numbers of microorganisms, but also if there's any changes to the diversity or if there's any changes to the microbial activity. And as I've mentioned earlier, qPCR gives us total numbers of bacteria and archaea and also numbers of functional groups of microorganisms associated with MIC, and ATP gives us the microbial activity and how that has changed. Next slide, please. The other important consideration when performing these tests in the lab is the reactor type. There are different reactor types that are available to choose from for MIC testing. So that could be like stirred batch type reactors, recirculating loops like Robin's device, or stirred continuous recirculating loop reactors like CDC bioreactors. Batch reactors, as the name suggests, are like these simple bottle tests with a coupon or multiple coupons suspended in a bottle with process fluids or any other sample. And in the Robin's device, coupons are flush mount in the device and to the flow, and fluids are circulated from a reservoir continuously. And once the fluid starts from the reservoir, it passes through the cell where coupons are and then goes back to the reservoir. And in a CDC bioreactor, coupons are suspended in bioreactors that is continuously stirred, and again, fluids are recirculated from a reservoir. Depending on the field conditions that we want to simulate, the type of test setup can be selected. To simulate let's say a produced water pipeline, a continuous flow through system will be most representative. For simulating the case of hydrotest waters that have been left in a pipeline for long periods of time, maybe a batch system or recirculating loop with low flow can be chosen. And biocides can either be added to the process fluids before exposing to the coupons, or they can be added after biofilms have been formed, depending on whether biocide testing is done to investigate effectiveness to prevent MIC, or if biocide testing is done to investigate if it can mitigate MIC. And in addition to these examples of reactors, that you can also use continuous once flow through systems, but they require large volumes of fluids and hence are not used as commonly. Next slide, please. Biocide effectiveness testing is the next step, and this effectiveness testing is usually done by investigating the effect of planktonic cell numbers, or by looking at how numbers of maybe common MIC-associated species, functional groups like sulfate reducers, acid producers have changed. But because MIC is mediated by biofilms, and analyzing the effects of biocides on biofilms and their and other parameters like corrosion rates, corrosion products, and chemical environment is important, a more comprehensive testing is recommended. So this more comprehensive testing would look at if a biofilm is controlled in addition to a reduction in planktonic numbers, what the effects of the biocide are on the biofilm and planktonic community composition and activity. So not just looking at how the numbers have changed, the abundance, but also looking at if there's any changes to the composition and if there's changes to the activity. And more importantly, because biocides are applied to mitigate MIC, it is important to investigate if general corrosion rates and pitting corrosion rates are reduced. Most often, this is a step that's missed in all of these biocide testing that is done in the lab, where the focus is maybe only on planktonic population or the biofilm population, but not on corrosion. But if we are applying biocides to mitigate MIC, we should not forget that corrosion rates and pitting rates are important to also look at. The biocide should be tested to see if there's a possibility of biofilm regrowth, and changes to fluid chemistry after biocide has been added should be investigated to see if there are more nutrient sources to the microorganisms, and whether biocide addition and whether after biocide addition there are indicators of abiotic corrosion, meaning MIC has been mitigated, and now all you're seeing is indicators of abiotic corrosion. Changes to dissolved gases is important to look for, and compatibility issues between process fluids, biocides, and other process chemicals it's also important to consider. The effect of process chemicals like drag reducing agents and oxygen scavengers on biocide effectiveness and mix should be tested. Any visual changes to the coupon in terms of deposits or corrosion products is also important to consider, because that can also help understand the corrosion mechanism and if there are any other changes that are happening in the system. And once all of this information is collected, this data should be integrated to infer about the effectiveness of biocides in preventing and mitigating MIC. Next slide, please. So to summarize, a number of different variables influence biocide effectiveness and can be accounted for during these lab tests. So it's important to integrate data from microbiological population and chemical and physical environment, the operating conditions, materials, and corrosion products. And also to mention, planktonic kill tests are not enough to determine biocide effectiveness, and using advanced microbiological methods like ATP, qPCR, sequencing can improve our understanding of the effects of biocide. And these techniques, as I mentioned, should be used along with information on corrosion products and corrosion rate monitoring to evaluate biocide effectiveness. Now Rick will give an overview on an upcoming joint industry project on MIC.
Great, thank you, Susmitha and Chris. Just briefly before we go into questions, I wanted to mention this joint industry project that we're currently working on with 2 technical partner companies, one being Microbial Insights and the other being ExxonMobil Upstream Research Company. We're currently putting together a JIP on development of water-based advanced MIC detection and monitoring technologies and biomarkers for MIC and associated KPIs. So we're looking at a multi-year project, probably on the terms of 3 years, using produced waters from a wide range of oil field operations, or hopefully around the world, particularly those that have severe MIC issues and a history of MIC issues. We're going to have an opportunity to use some very innovative test equipment that's designed and constructed for biomarker discovery, using actual pipeline conditions, including acid gases and pressure and temperature and so on, chemistry. And really, we are looking for expertise and knowledge from industry experts to join the team to be developing this technology to move MIC to the next level, basically, to move MIC diagnosis to the next level. If you recall, Chris and Susmitha were talking about how there's not one single isolated indicator that can tell you that MIC is occurring or not. It takes integration of information. We think that the next level for MIC diagnosis is going to be using advanced biomarkers, such as already have been mentioned by Enning et al. So yeah, it's going to be an exciting project. If you have any interest in this, you'd like to hear more about it, you can reach out and contact me by email. Also, there is a, you can reach us through Microbial Insight. They have a web page that's shown here on the JIP. So thank you very much. And I think we're going to go to questions now.
All right, thank you everyone. That was a great presentation. So the first question that we have is, what indicator or collection of indicators in the lab simulation shows us whether the biocide application should be continuous or done in batches?
So yeah, that's a great question. I think a lot of times when we think about that question, it mainly comes down to how it can be applied in the field, and what makes sense. I know we've looked at both, and what we've seen in the past is, say you have a biofilm that's established on a surface, and most of the time in a pipeline, if you're going to do a batch treatment, say you have a flowing system and you would apply a slug of biocide. So this biocide can go through the pipeline, and then your biofilm will be exposed for a short period of time. What we see is that there might be some immediate short-term effects on the biofilm that's present. But then within usually a day or two, things kind of go back to where they were, because if you can think about it, after that biocide goes through, you're probably going to have some fluids, or whether it's oil or water, that contains all of the same microorganisms that were there before, just comes right back through. So any benefit you see typically tends to be short term. Batch treatments kind of make more sense if you have kind of like a static process. Say maybe you're treating a tank or a vessel where things are going to sit for a long period of time, and so batch treatment would give you a longer term continuous contact with the bacteria or microorganisms that are present on the film. So that's usually what we consider when we think about batch or continuous treatment. From the things we've seen, the longer you can expose microorganisms to a biocide usually has a better effect. But sometimes you're limited based on economics of the situation or operating conditions or any number of factors. So there isn't really a single indicator in the lab that we look to say which to use, but it ultimately comes down to effectiveness and how you're affecting corrosion rate. Rick or Susmitha, did you guys want to add anything to that?
Yeah, I wanted to quickly add one thing. So depending on, let's say if you only wanna, or if you're only able to use one application mode, we can always set up these experiments in the lab and see what concentration can be added to reach that effectiveness that we are looking for. Or if there's a question on which mode to pick, and we can, as mentioned, we can always set up these tests in the lab with one mode of application and compare it with the other mode of application to see which application mode is more effective.
Okay, great. Next question. Do you have any idea on how microbial adaptation to biocides contributes possibly to MIC even over time?
Susmitha, do you want to answer that one?
Yeah, there is a lot of literature out there saying that microorganisms may start developing some sort of a tolerance towards biocides, or microorganisms can go into, like when microorganisms are in a biofilm, they can exist as persister cells. So they may be in this dormant state where they're not taking in biocides. So whatever biocide you're adding is not as effective in killing these microorganisms because they're not taking in the biocide that you're adding. So, or there are some instances where microbial tolerance to biocides and these chemicals has been shown. So yes, that is a concern, and which is why doing these tests in the lab before biocides are applied is important. Rick, do you want to add anything more to that?
No, I think you did a good job. I think that's why we often emphasize if it's possible to do mechanical cleaning too, because that just does so much to help break that biofilm down, before you're applying your chemical, and trying to avoid having those persister cells hanging around in the biofilm. But like you said, Susmitha, very, very well stated that it's not necessarily resistance that we're seeing, but it's just kind of an adaptation of the community to the existing conditions. But
I think this next question is a good complement to that. It says, in your experience, what's the impact of biofilm density on the effectiveness of the biocide or inhibitor? Is it something that you stimulate or you can simulate in the lab? And then the second question is, oh no, same one, just a repeat. So yeah, so can you simulate the biofilms in the lab, and what effect do you think it has on it?
Do you want to start with that one, Susmitha, and I'll add on?
Yeah. So when we establish biofilms in the lab, some of the things that we can look for is also like how biofilm viability has changed, biofilm thickness has changed by looking at by using microscopy-based methods. I think that's what you mean by density, or I'm trying to understand what exactly constitutes biofilm density. It could also be the amount of cells that are in a biofilm in addition to the thickness of a biofilm. So yeah, we can use microscopy-based methods to evaluate how biofilm thickness or density has changed, and there are image processing techniques that you can use to monitor how biofilm characteristics have changed upon application of biocide in the lab.
Yeah, so one thing to add to, so we've seen to where shear rates influence kind of the biofilm thickness that forms up. And so the higher the shear rate tends to result in a more compact biofilm. It's still the same number of microorganisms kind of tend to end up in that space. It just kind of changes the thickness and density of that biofilm. So that's why one of the things we want to consider is the shear flow rates that might be experienced during normal operations. And then so we can set up systems with cells that either have a flow through system and we can mimic that shear rate, or if we do kind of like the bottle test as Susmitha showed, we can change the stir rate and come close to that the shear flow that you would see in operating condition, and then that can influence your biofilm thickness. They tend to reach a steady state, but what we see is that the ability of a biocide to penetrate that biofilm, they tend to be pretty good at resisting chemicals and other things from penetrating them. So what we see is that there's kind of a limited effect, and it just tends to affect the surface microorganisms and not really penetrate deeply into the biofilm. So that's why we use some of those techniques like microscopy to take a look at the film and see what's present. Or you can use live dead staining to also get to similar assessments of what's going on your surface. But yeah, typically through control of flow rates and shear, that's how we try to control our tests and biofilm thickness. So hopefully that answers your question.
Hey, Chris, I was just going to jump in and add something quickly. Another thing that we see that has a big influence on that, though, is not just density or even cell numbers, but it's composition, such as from inorganic species like corrosion products and scale, minerals, and things like that, because that all gets bound up in the matrix of the biofilm, and that offers some protection and some mechanical integrity to the biofilm too. So that it doesn't take much biofilm thickness to accumulate a pretty impenetrable layer on the surface of a material. So yeah, one more thing I wanted to add to that. Thanks.
Oh, good point.
So do you guys correlate the biocide dosage to the biofilm thickness? And do you find that there are certain types of biocide that's better at penetrating the biofilm than other types?
Dosing, we don't typically correlate to biofilm thickness. So we usually just apply like a range of concentrations just to see what would happen. And I don't know that we've seen any correlation between the two. Systems, especially these living systems, are so different that it's hard to predict performance just based on one single criteria. So we haven't seen a correlation between the two. And then regarding effectiveness of chemicals to penetrate biofilm, I don't think we've seen one yet that has really, well, I guess what we'd say is like been a very effective at penetrating biofilms. Again, it depends on what's present in your biofilm. If there's things that form this extracellular polymer matrix, those tend to be more resistant to biofilms or to chemical penetration. But I don't know that I've seen anything that is as effective as, say, mechanical cleaning at removing a biofilm.
Would it be correct to assume that the higher the shear rate, when you were talking about shear earlier, that would be the lower the effectiveness of a biocide could be on the system?
I don't know that I'd want to draw that conclusion necessarily, just because there's so many variables that go into it that I wouldn't want to overstate any one aspect. But a higher shear rate should lead to a more dense biofilm, and the more dense it is, like the harder it should be for a chemical to penetrate. So I would agree with that thought process, but I wouldn't bet my house on that, since it's hard to, without the data, I wouldn't want to come to that solid of a conclusion.
Sure.
I just wanted to add to that. I agree with what Chris said, and there have been some studies that have also shown that with high shear or high flow rates, the biofilm thickness is smaller, but the biofilm is more robust. So maybe we can say that it's harder to remove the biofilm, but again, we do not want to generalize that.
Okay, the next question, do you guys find that microbial attachment is influenced at all by the ionic strength of the system?
Rick, do you want to tackle that one?
I don't think we've seen a clear correlation between ionic strength. It tends to be, the more you get toward not extreme conditions, the more diversity that you can have in the environment, and then the more options you have for biofilm robustness. But I think we've seen in some pretty high salinity situations where we still have some biofilm that's still challenging to deal with.
Yeah, I think in general we've seen very few instances where we haven't been able to grow biofilm. So biofilms seem to be pretty easy to form, unfortunately.
Shows the protective nature of the biofilm formation itself in these systems, right?
Mm-hmm.
Okay, next question. Is it common for you to test combinations of biocides as opposed to just testing a singular biocide?
Yeah, we've looked at that scenario, and again, like it's pretty simple with a lab setting because you can control a lot of your variables. So we think that's one of the advantages of doing laboratory testing is the ability to evaluate that and see, how does a combination biocide compare to each individually or different biocides. So that's one of the more useful tools here is to screen and evaluate something that would be more difficult to do in the field.
So in one of your earlier slides, you showed a sample set where you were looking at iron reducers and iron oxidizers, and they were both detected in the system, even though the water seemed to be pretty reduced. Can you elaborate on what you think caused the presence of the iron oxidizers in that system?
Susmitha, do you want to take that one?
Yeah. So the snapshot that I showed of both, those are just representative samples. I don't think they were from these were not exact sample sets. We did not want to put the actual values in here. So very commonly we do see different sorts of microorganisms in the sample. We see iron oxidizers, iron reducers, and others too. And what we do is we characterize what is present initially in the sample, and then we also look at what's present in the end and what's forming on the biofilm, and then we do comparisons and then correlate that to water chemistry. So that gives us an indication of how after addition of the biocide things have changed. But to speak from what is there in the sample to begin with, it's usually more complicated to understand what might be causing the presence of a certain group of microorganisms. There could be a lot of upstream processes and other samples that are coming into the system that might be sources for particular group presence of a particular group of microorganisms.
Okay, great. Can you, oh, sorry, go ahead.
I was just going to say one thing too to keep in mind with qPCR is that it's both things that are living and dead. So that's why we combine some of the things with ATP is just because there's something there. It doesn't mean it's active or participating in whatever mechanism that you're seeing. So.
All right, when you guys do a site evaluation, can you talk a little bit about the typical cost of a study, how long it takes, and then how do you translate that to field success then in the end?
So a lot of it depends on scale and level of involvement and how much data we want to collect. So I guess there's varying tiers of analyses that can be performed, but and how long experiments need to take. So every scenario is different. We haven't seen any, we haven't had a lot of repeats of the same conditions and questions to answer. So it's difficult to come up with a single cost. And so it's kind of unique and tailored to each person's specific needs. And we try and work with our clients to make sure that we're coming up with the most cost-effective solution for them, to ensure that we can answer, the relevant questions that can get them started. I guess what we've seen is I guess we've been able to identify some chemicals that we think are more effective than others, or been able to provide recommendations on which doses, if an increase in dose would be more effective than the current treatment, or if it would be possible to reduce a dose based on laboratory testing. And then following up that with monitoring in the field, I think we've seen similar results to what we see in the laboratory. Rick and Susmitha, do you guys want to add anything?
Susmitha, do you want to say anything?
I think Chris did a good job summarizing everything. So yeah, we do these tests in the lab, and then we can always, based on what level of information is needed, we can tailor to like a quick and dirty screening method, to somewhat of a more detailed comprehensive testing, to a full detailed screening and optimization study. So we can do a wide range of tests depending on what information is needed, and based on what information is needed, we can pick out the test that we want to perform in the lab.
Yeah,
Rick, do you have anything to add?
No, I think well said. I think no two situations are the same, and I think we do like you both said, we do a good job of trying to accommodate, the test protocol to the customer's question and their resources to answer the question. In some cases, if you're making 1 million barrels of oil a day, money's not an object, right? But if you're making 99% water and 1 barrel of oil a day, then yeah, it's a big problem. So yeah, we just try to work within that and do the best we can. And the other thing to add is just that even though we do these tests in the lab as kind of screening, optimization methods, there still needs to be field proof that this is actually working, and we try to help our clients by following up with recommendations for that field monitoring as well, to prove that it's actually working.
Okay, last question, you guys. What are the common indicators that if there's been a history of MIC issues in a system that you would look for that would trigger the start of a laboratory simulation study? Are there a couple indicators that you guys look for there?
Probably a history of leaks or uncontrolled corrosion. That I think that's where we tend to get involved, is the more challenging situations where maybe you're applying a biocide or corrosion inhibitor, and you just see nothing is changing. Your corrosion rates are not good, not where you want them to be, or you've had a leak that you just didn't expect. That's a lot of the times where people call us, but I think it's we've started to see some more proactive approaches where before even applying any chemical, just even considering it, if you have a corrosion rate that for your system is problematic, what are the right things to do to mitigate that corrosion? And so that speaks more to the corrosion management process than any sort of laboratory testing. So that's kind of the point where we like to get involved, is before applying any chemical or considering chemical, kind of doing a holistic approach to corrosion management, and using chemicals as it's kind of like the last resort. But that's typically what I've seen. Susmitha, do you have anything you want to add?
Yeah, there are some other instances where we've been involved where we are comparing different biocides from different companies, and then we try to do that independent evaluation of which chemical gives the most effectiveness. I think that's one case where we've been involved.
Rick, anything we missed?
Nope, I think we're good. I think we could put a wrap on it.
All right, thank you guys so much for answering all the questions, and thank you everyone in the audience for attending. We know you're busy and we appreciate that you took time out of your schedules to be here with us today. And Rick, Chris, and Susmitha did a great job on this presentation, and we really appreciate you guys taking the time to be with us and DMB's partnership with us over the years. We really enjoy working with all of you.
Thank you, Dora.
Yeah, thank you. Thanks everyone for attending, and thanks for your time and excellent questions.
All right, thank you everyone. And if you have additional questions, you can always send them. You see their email addresses on your screen here, or you can email us at info@microbe.com, and we'll be happy to get with them and get the answers and get back to you. So I hope you all have a wonderful, safe day. Thank you.
Thanks, everyone.